First thing I wanted to do was to just set a couple of ground rules. One, there will be no app-based questions as a result of a glitch in the app. So we'll just do some free mic. Please raise your hands if you have questions as things go through. The sound apparently is not particularly good at the back of the room. Although the mic is on, I can hear a bit of resonance. Okay. Is that better? Back of the room? No? Just waving at me in general. Okay. Long time no see. The other thing I'd like to do is obviously to congratulate one of our speakers, Lotte, Bjerre, and Knudsen, on recently being announced as one of the winners of the Lasker in conjunction with our own Joel Habner and Svetlana Mojo. So, a fantastic achievement, and I think really sets the scene for today's discussion. Our goal is really to explore the entire cardiovascular space and what it is about chronic kidney disease that requires reimagination of the pipeline and how that pipeline makes it into the clinic. And so we've got a really broad-based panel, which I hope will allow us to flesh out a lot of the different components of this. I'm going to ask each of the speakers to give a brief bulleted introduction. Once I ask the first question, I'm co-chairing with Jason Gerberry from Bank of America, who will also be doing cross-examination under certain circumstances. So the first thing I'd like to do is to ask Deborah Wexler if she can really set the clinical scene for how cardiovascular disease has really become so much more than it was even when she and I were at Mass General many years ago together.
Hello, everyone. I'm Deborah Wexler. I'm the chief of the diabetes unit at Mass General Hospital. I'm an endocrinologist, clinical researcher, mostly working on how do we take what we know works and make it happen in the real world, so translation to practice. I think to be an endocrinologist in a cardiovascular disease panel is, on the one hand, exactly where I think I should be, because of course we as endocrinologists think, hormones control the whole body, but the fact that I am here speaks to what a paradigm shift that we have lived through. So when I started using a lot of the medications that are now considered cardiovascular medications, no one really thought they were cardiovascular medications. GLP-1 receptor agonists, SGLT2 inhibitors were glucose-lowering medications, and there might have been a hint that they might do something else. But really, that is how they were designed, and that is how we use them, starting really in 2006, 2007 for exenatide, and then in 2014 for SGLT2 inhibitors. But the rapidity with which they have changed the cardiovascular disease management paradigm is striking. And so I feel I'm where I should be now on this cardiovascular disease panel, but it's really been quite striking. I don't know if that kind of captures.
No, that's perfect. I think really the reframing of the entire field, not as cardiovascular, but really as cardiovascular, renal, metabolic, wellness, obesity, the blending of all of these chronic disease spaces is really very obvious, has been, I think, clinically for decades, but biologically and pharmacologically has not really emerged. Lotte, you obviously foresaw a lot of the promise of GLP-1 manipulation many years ago. Did you imagine then that it would have such diverse implications in so many different disease areas?
So we actually knew about it fairly early. So we knew about the potential in obesity also all the way back to the 90s. Obviously, we needed some time to show the clinical benefits. And also around the year 2000, we started to have the first biomarkers for cardiovascular disease that were positively affected with GLP-1. I think what's difficult for some people to understand is because it has effect on so many biomarkers at the same time, and by the way, differently than SGLT2 inhibitors, so it's great for the patients that these classes actually work in a different way. So we had all the way back from, I think the first paper we have out is from 2008 with dose-dependent reductions in both CNP, ICAM, VCAM, systolic blood pressure, and triglycerides, just to mention a few. So it does go back quite a long time.
But when the drugs were being developed, was it clear as you were building development and implementation plans that you would need to really reimagine where they were to land?
Yeah, so I guess we've been trying to get towards the cardiologist for quite some time, but it really took time. And of course, we realized that, first we had the cardiovascular outcome trials in people with diabetes. Then people were saying, oh, but it's just glucose. And then we have the cardiovascular outcome trial in people with obesity and no diabetes. And then now people are saying, oh, it's just the body weight. But it's actually not, because we have mediation analysis that shows it's not the body weight. It's probably more inflammation. And then, of course, we realized that we had to do many more trials, and that just took a long time. But now where we have also both the HFpEF and HFrEF data, it's super interesting, Mike.
Absolutely. Christoph, you've obviously led a lot of drug development and invested in a lot of drug development through your lifespan. How do you see us matching the clinical scenario better at early stage development? How are we going to avoid taking a decade and a half to really understand where a drug fits in the therapeutic space?
Over the last 20 years, I've started 7 companies as CEO that we took public out of Harvard and MIT. Those include Alnylam. And I am smiling at Lotte because Alnylam I founded as CEO in 2001. And it took us 15 years and $3 billion to get the first drug approved. And we never had any idea that 3 of our drugs that are either approved or the last one is about to get approved would be in cardiovascular metabolism. The first, inclisiran, for cholesterol, PCSK9. The second, an angiotensinogen RNAi therapeutic, is actually late stages, about to get approved for blood pressure. And the third, and that's the craziest story that we had no idea about, is for amyloid heart failure. Of course, first everyone thought it would be related to those who have mutations. And only way later, really smart cardiologists like you figured out with technetium scanning that perhaps a very high percentage of people with impaired cardiac function later in life have disordered amyloid deposition. So that actually will be probably our biggest drug. So I like to say chance favored the prepared mind. You knew, I'm sure, early on with GLP-1, but for us, it took us a long time to figure it out.
Well, it certainly took us a long time.
So Dave, you obviously are investing in early stage companies. Your companies are not doing $800 million outcome-driven trials. How are we going to accelerate the matching of, I suppose I'm asking the same question again, the matching of indications to perhaps clinical scenarios not really emergent, maybe until 10 or 15 years later?
Well, I think when we anticipate how we'll deploy capital to create these new companies based on transformational science across the globe, one of the things we're thinking about in being in the skate to where the puck's going to be business, it's a tough business to be in, and there are not a lot of people other than Wayne Gretzky who can really do it consistently. You have to start thinking about what's the differentiation that you can achieve on the time frame and the capital that you happen to have, right? And so one of the reasons why, sort of back in 2021, when we first had some high-dose data with Sema, and we were contemplating what the incredible transformation was going to be looking at weight loss data, we started thinking about new products that can have a TPP that's differentiated. And from that perspective, it was, what are novel pathways to think about? So in 2021, we got involved in investing in a company looking at the activin receptors to A and B, and looking at what might be differentiated with myostatin. You have myostatin, activin, GDF11, and others. So looking at a novel pathway that can have a differentiated profile whereby you're, keep maintaining lean body mass and perhaps improving and increasing muscle and decreasing fat mass, albeit maintaining some of that metabolic benefit. And importantly, what might be combinable with GLP-1. So what might differentiate that both from a tox profile for combinability and also a novel profile. So looking at those sorts of things as early as possible, I think really helps. Not all of us have the insight to do the research that Lotte is doing over decades and have a clear North Star for how you're going to develop these things. And by taking sort of focus bets, looking at some of these endpoints, I think that's a very important thing to do as we explore where the novel pipeline can come from. We're talking about Renewal. Right now we have 79 clinical programs or something across, I think it's 52 companies looking at 41 targets. So there's a lot of redundancy already in the pipeline. So trying to find what will be differentiated will be very important. And then to your point about trials, one of the things I hope that gets uncovered by all the incredible work that Lotte has done at Novo and Lilly, and Deb is leading the way here, is to try to understand new phenotypes of what we're talking about here, right? So unfortunately, we're still using a device discovered in 1881, the sphygmomanometer, in how we diagnose hypertension. And so can we find better ways, whether that's using deep learning, machine learning techniques? Can we get comfortable with non-hypothesis driven ways of identifying novel phenotypes? And I think that will ultimately help us carve out faster paths to what may be beneficial in patients sooner, and therefore you can sort of put more capital and more effort and energy behind them earlier, knowing that there is a higher probability of success.
Those are, I think, excellent points. I mean, you know how much I love my medieval phenotypes. But going back to Deborah, in a related angle, how are you thinking about exactly what Dave just mentioned? Stratifying patients potentially on the basis of drug responses, but importantly then positioning multiple therapies. We're not going to see, I don't believe, as Lotte pointed out, triglycerides. How many $800 million trials have there been for triglycerides, none of which affected outcomes? And guess what, if you use GLP-1 RAs, triglycerides almost disappear as a problem in every tissue, as well as in your circulation, and you're getting all of the outcomes. How are you imagining we're ever going to redesign guideline-directed therapy as we move forward when there are all these new compounds coming in a space that really, as we've said, is complicated and has overlapping phenotypes that are poorly differentiated?
So I think, being on the clinical end of the spectrum, the question of how important the phenotypes actually is I think an open question. And what I would say is that in type 2 diabetes, there's enormous heterogeneity of type 2 diabetes. And I, as a diabetologist sitting in the clinic, years and years ago, said, we really need to understand the different phenotypes, the different heterogeneity. I remember even putting forward a proposal to look at these phenotypes that indeed were subsequently borne out by the genetics. So my colleague Mary Mudler has looked at this and is able to cluster sort of liver diabetes versus insulin deficient diabetes and all of those new phenotyping schema that exist. I think as a diabetologist, as people designing drugs, we want to rationally target the underlying pathophysiology of each type, subtype of diabetes, or perhaps subtype of heart failure. But yet it is striking that the drug that works for one phenotype, as a practical matter, frequently works for all the others as well. So SGLT2s work for HFpEF and HFrEF. In diabetes, many of our diabetes drugs really work for all diabetes phenotypes. They may work better for some than for others. There may be gradations there. But as a practical matter, it may or may not be that important, I wonder. And I do think as a practical matter on the implementation end, it has become enormously confusing for clinicians on the front line to know how to identify these subtypes. And so that has been a real challenge. So I think when we've seen pharmaceutical companies target rare diseases where they know they can go to specialists who manage this condition, who can deliver that drug, that's one avenue. But when you're thinking about a population-wide condition like hypertension, like type 2 diabetes, like coronary artery disease, is it more important to get a good therapy out there to everyone or a targeted therapy out there to a few, getting that phenotype exactly right? I don't know. I think that's an open question.
I think it is an open question. I think it's a great one. Lotte, as you're seeing the future, for example, of GLP-1 RAs and all of the associated pathways, do you think they're going to displace statins? Are they going to displace PCSK9 inhibitors? And if so, how are you going to understand that? How are you going to learn it?
So I'm not even a medical doctor, right? So I'm not sure.
But biologically, though.
But maybe I could say something, because I'm a big believer in data, right? And I think that actually with modern technology and using omics more, I think we actually could learn a lot that then could be transferred also to more individualized patient care. Because I think we are moving towards a future where more people would actually like to have their own data used to inform trials better and inform their own treatment choices better. And I think about 6 months ago, I even saw like a commentary from someone from FDA, I can't remember who it was, but said, let's get some more omics data on. They were singling out semaglutide because it's so widely used, right? But let's get some more omics data, because I actually think we could learn a lot with all the data that's available. And Zuckerberg and Chan, they're building the human atlas, right? So I think in a few years with AI also and machine learning, I think we could actually be, I'm hopeful, we could be actually having a much more informed decisions.
Perfect. Lotte, you used the AI word. So yeah. We're going to put that aside for 1 minute. We'll come back to it. Jason, you had a question.
Yeah, just a quick question, Lotte. We see a lot of companies and investors struggle, I think, with looking at novel targets in the cardiovascular space, and there's obviously going to be a large outcomes trial that's required, right? So these companies have interesting data on biomarkers and surrogate endpoint data, but it doesn't really answer what's going to be the event rate of this therapeutic. So curious, as you guys developed GLP-1, how you navigated and leveraged early data, right, to make such large-scale investments in outcomes trials. And is it in the end just a leap of faith? Big market, you're doing a lot of good things, and a leap of faith that there's going to be an outcomes benefit.
So obviously the first one we did was the LEADER trial with liraglutide and diabetes. That was kind of forced by FDA and EMA, right? It's not that we didn't want to do it, but still eventually it was forced because of what happened with the Knudsen analysis and all of the diabetes trials. But the next one SELECT was an informed decision. There, we believed in it. We didn't know the mechanism, but we had all of these biomarker data. So that was, you could say, was a leap of faith, but it was a leap of faith based on a strong belief based on data.
Christoph, you're obviously also thinking long-term about your investments and trying to understand how compounds are getting into the clinic. What's your take on how either precision medicine, as it's been defined in the last few years, although it probably was molecular medicine 30 years ago, and a whole host of other things in between. How do you think that's actually going to end up getting to the clinic? Or is it, as Deborah said, maybe a false premise? And do you factor that in as you're building a portfolio of companies going to the clinic?
I think the neat thing is that the game that David and I are in is a different game than the game Lotte is in. We're in the very capital-constrained startup biotech world where we have to very quickly try and zero in on a specific mechanism with an extraordinarily high effect size. What's interesting for me is to watch those worlds come together because actually we would have never predicted that our drugs would be starting to be used in primary care type indications. But I think what we've learned once you understand a genetic lesion, and with RNAi, it's a perfect setup. The p-values we generated in some of our early studies were so extraordinarily tight that you could go into primary care indications and you could contemplate doing larger populations because you would see such significant effect sizes. That's something we never would have guessed, but actually it's really beautiful to see it developing.
And for example, to take a couple of distinctions that you made there. So, hypertension, obviously the effect sizes of the renin-angiotensin system are tiny at a population level, 1.2x. Amyloid is huge, it's binary, it behaves in many instances as an autosomal dominant condition. And yet I see across the spectrum, I see people investing roughly the same amount of money in effect sizes of 1.2x as I see in effect sizes of 1,000x. So is there actually a rationale in the way that people are thinking about investing, and based on the biological importance, or as Deborah said, is it really just a matter of you pick your condition and you find a vertical and one pathway that works in that condition, and then you build that as your entry into the space?
Well, Sidney Brenner, who won the Nobel Prize, said we should not focus on making the measurable important, but we should focus on making the important measurable. And so I think what we're starting to find is if we can look at genetic lesions and really define patient populations, sometimes we're lucky that more patients than we expected on a population level have that lesion, and those are the drugs that we can get approved.
Got it. Jason?
Yeah, just curious, Christoph, the question about precision oncology style medicine in the cardiovascular realm, at least in precision oncology, we saw a lot of capitalization occur in oncology over the last 5 to 8 years, but we also had a surrogate endpoint of tumor shrinkage that I think was a definitive proof point, whereas in oncology, in CV, the traditional endpoints, there's no getting around them, right? I think with Alnylam, they tried to approach 6-minute walk tests with TTR silencing, and the FDA shot them down, and they needed an outcomes trial. So as much as this sounds interesting, I just wonder how, from an investment standpoint, you think about that.
Well, we could have never, and I know David probably feels the same, we could have never, as early stage venture capitalists or founders of companies, contemplated those kind of studies. We, when we're starting these companies, have to take something out of Harvard and MIT and try and train it on a specific genetic lesion in a certain population. Then later, I think it's these translational medicine people that I'm lucky to be sitting next to who figure out, oh, wait a second, it can be used in these unexpected heart failure situations where maybe 10% of them have amyloid deposits. It's kind of the cool thing. I don't know, David, what you think about that.
Yeah, maybe, I think Deborah made a very good point, and we're trying to figure out where are phenotypes important, where are they not,
Yeah.
and certainly we'd all rather treat very big diseases with big effect sizes. But to parse it out, if I think about something like hypertension and then maybe bring it to something like preeclampsia, right, where we've made tremendous progress in understanding the pathophysiologic nature of that disease, when you've got just this background, 130 million live births, you've got 2 to 8% with preeclampsia, you've got 5 to 10 million women across the globe with severe complications of pregnancy. And we just used to say, well, hypertension and maybe you're spilling some urine and you have proteinuria, maybe we're clustering that, but now we seem to know that soluble FLT1 elevations and its ratio to placental growth factor is a really important determinant in diagnosing, and may also be the factor that is causative pathophysiologically in that disease population. And so what's interesting is if you can go from just hypertension to very specific molecular basis and a cluster perhaps of phenotype, but what early stage biotech is attempting to do is take that physiologic understanding and match it to the best modality. And we're living in this golden age of how we can deploy genetic medicines. There's some talk about siRNA, and I just credit John Maraganore and Akshay Vaishnav for reducing to practice siRNA across 6 different approvals, and we're now the beneficiaries of that at a new company that's looking at siRNA to block soluble FLT1 from being secreted from the placenta. And again, if you think about which modality am I picking to hit this target, small molecules, you don't want to get into the fetus. Antibodies, very tough to make the soluble FLT1 because it's just got such high homology to other proteins. And so SI turns out to be the exquisitely precise drug that you can utilize in this setting should you be able to get it into the placenta. And so it's a long-winded way of saying, if we think about hypertension, we think about subsets that might be benefiting from, our novel insights on both the disease pathophysiology and all these new genetic tools and modalities that we have to bring to bear. I think you can get, in many instances, a lot of progress both on large indications, dyslipidemia, for example, as well as some of these more discrete ones where you're affecting populations, more precisely.
So to distill what you and Christoph both said, I think the devil is in the details when it comes to individual mechanistic insights, but I'm also hearing from both of you a sense that actually the real advantage in rare disease is that it actually gets you a better regulatory pathway. That ultimately your goal is a rare disease that has a land and expand type possibility in a much broader syndrome. Yeah. Is that a reasonable?
Yeah, I think that's absolutely true. Although I would say in the context that we're talking about, the cardiovascular disease outcomes, CVOT trials may not be required of small companies, but they may be definitive as far as being competitive in those indications. So you might get regulatory approval based on something else, but you may not get the kind of real-world utilization without larger outcome studies for
So I think that takes us on to sort of another theme, which is how do we think, and Jason, you highlighted it in your reference to the oncology framework, how are we going to redesign this sort of what seems like an almost impossible set of barriers other than through serendipity to systematically get drugs into the right place with the right amount of investment at the right time as they're moving through the clinical spectrum of utilization. I think you're going to see this, for example, Lotte in GLP-1, as the prices change, as orals become available, as people are thinking about combination therapies. Are you going to end up diffusing the entire field, or is there going to be a really systematic set of maybe, Deborah, real-world pragmatic trials, adaptive design? How are we going to rethink how something as common as cardiovascular with such non-approved biomarkers as we've described, in other words, non-approval feasible biomarkers as we've described, how are we going to reimagine that space? I think I'll ask you to go first, Deborah, and then ask Lotte for her insights.
I'm not sure there was a lot of questions during the question.
That's how I roll.
That's how you roll. I mean, I think what is interesting, what we see a lot is, okay, now that a therapy is out there, how would an investigator, a practitioner in clinic, having an insight, then take that into a trial? It might be a pilot trial. An example of this would be SGLT2 inhibitors for the treatment of gout, for example, which I think has been not necessarily driven by the pharmaceutical companies, but by investigators managing patients with gout who say, oh, gee, we really need to test this for this indication. And so a lot can, I think, be diffused that way in smaller trials. And once it's approved, of course, the bar is not as high. I just want to highlight, come back to one point that you said, which is, it wasn't that, and if you could clarify this, it wasn't that Novo Nordisk said, oh, okay, this drug is going to be good for heart disease. It wasn't that the people who make SGLT2 inhibitors said this drug is necessarily going to be good for heart disease. Those trials were mandated as cardiovascular safety outcome trials. And maybe you could clarify whether you guys thought there was going to be a benefit, because my understanding was that it was a little bit of a surprise.
No.
Okay, so explain.
We powered LEADER for superiority.
You powered it for superiority.
Yeah,
so you had an inkling that it would be?
Yes.
Okay. But the
Right,
mandate,
it was
the
mandated,
mandate was
but we
for safety
powered
though.
it
So I think,
theoretically.
so it's just an interesting thing to think about how that discovery, which really was transformative, kind of came about.
Yeah.
But it is stunning to hear just how long it takes
Yeah.
to go from an initial idea in a vertical that sounds like it's really relevant across the board to actual definitive indications for use. And then we haven't even gotten there yet, but we will go on to thinking about implementation because we still have to actually teach people how to prescribe it. Lotte, as you said, you saw this in the future. Did you see any other indications that are not currently being tested in your future scope?
I'm not sure I dare say that in a room full of medics, right? But there's a lot, but it's not proven, right? So that's very important. I think we have the kidney benefit also proven now. There are good hypotheses for neurodegenerative diseases, but we don't have any good data. We are in Alzheimer's phase 3 with 4,000 people, but of course that's a high risk, not risk for the patients, but risk financially to see if that works. There's a lot going on around addiction, right? But again, it's not proven clinically. Can I say something about the previous one with the
Absolutely.
data also? Because it's been my dream for the longest time to actually have more data and then figure out how could we figure out what's going on with the risk we're not targeting, right? So we know that all the different classes of medicines, we know, it's typically 15, 20, 25% risk reduction. So what's with the other 75%, right? How do we get better at that? But I think with the tools that we have today, with much more in details and statistics, and again, back to the omics, I really love my omics, right? And the classical biomarkers to figure out, how do we get inflammation to zero? We know we can get LDL to zero now, thanks to you, right?
Right.
So how do we then say, how do we pair that up with all the data? Say, if we want to get inflammation to zero, what's the most important mechanism for that? If we want to get non-LDLs to as low as we think we can safely go, what's the most important biology to do that? And I think with the involvement of statistics also at the omics level, we can actually get much more informed about that. And then we can say which modality for which biology.
So I think the regulators have a fairly similar view of biomarkers and omics as they do of new drugs. And I think that's one of the problems is they're trying to actually show utility. What do you think? I mean, we've been talking about genomics. The technology's been mature for 15 years, literally completely stable for 15 years, still not in the clinic. Why is that? How are we going to change that if it's really the feedback loop for better drug discovery, drug development? Christoph.
That's a hard one. Everything takes 20 years in my experience, 10 to 20 years. So we're kind of right there right now, right? I mean, we sequenced the genome 20 years ago. I think it's just starting to happen right now. What do you think, Lotte?
Maybe with more compute power, but I thought it was super interesting to see NVIDIA when invested in recursion, right?
Yeah.
So with these kind of super AI power combined with deep biological insight.
I did not plant this question, but Dave, that's actually ends up being one of the core problems is you basically have 21st century technology going into, and I don't get me wrong, I love recursion, I love helix, but ultimately the clinical phenotypes are 18th century phenotypes, as we've heard. So are we going to reimagine how we think about the clinic and discovery and development being integrated? Because that sounds like, I think you're right, Lotte, it may be the way forward, but it doesn't seem like we're close to that at the moment.
It's a challenging one across the board, right? So if you think about some of the analogies from oncology that you mentioned earlier and that we're all trying to learn from, but even with that, right, we don't have BRAF clinics, right? We have thoracic oncology clinics. And so, we're still anatomically diagnosing and screening patients and moving them forward in that way. And so I think we're just starting to build into this, whether it's circulating tumor DNA or whatever profiles. But it's not, I mean, the kind of research Deb's doing is critical to figure out like how you can ultimately integrate novel delivery and ways of incorporating patients and getting them the right access at the right times, as well as making the benefits for these diagnostics that need to come on and be, I think, absolutely right. How do we embed more omics into these trials? But the throughput, the cost, and then I think the downstream implementation, we've got to make progress on all three of those things. And it is where if we come back to AI, we may get some better, more rapidly assimilated tools to help guide us in those endeavors.
Lotte, you had a question.
Yeah, it got me thinking, is equity in this debate, right? Because looking at black minorities and things like that, because I think that could also probably be really important to understanding some of the residual risk, right? Because we always struggle as a drug industry to get enough of different subtypes in our trials. I don't know why we can't do better, but it's just really, really hard. But I'm thinking to have actually a real strong focus on that and actually study them better, also with the differences with omics and genomics, right? I think that could be really important also.
Yeah, I think you're right. As a geneticist, I mean, most of the information content is in the extremes. Having extreme risk in your studies and thinking about it in lots of ways, including equity, is really important. Deborah, before we open it up for some questions from the audience, the last piece, I think it's come up a few times. You mentioned it earlier, Dave and Christoph mentioned it. How do we actually get these drugs into the clinic? How do we train non-diabetologists? You've had years of experience using semaglutides. How are we going to get primary care? How are we going to get cardiology, nephrology, neurology using these drugs and using them appropriately?
Yeah, so it's interesting that, not only do we not have BRAF clinics, we don't have cardiac kidney metabolic clinics. So the way that we're organized, we've often thought, we need to totally reorganize to target the underlying cause of this. So one is, do we need to really have multidisciplinary centers is one issue. The other question, though, is uptake. It's often said it takes 17 years for therapy to, have uptake, and if you count potentially the first approval of the first GLP-1, then we're right about 17. But if you actually look at the rate of uptake of the newer medications, it's been remarkably rapid, limited to some extent by cost, but I think limited less by practitioner knowledge, because I think people have now seen with strong effects that this is something that's worth learning. And of course, there's been such attention in the popular press that there's patient demand for it as well. So it's almost like this is a special case of uptake. Certainly in the past, when we've tried to teach people how to use new drugs, new therapies, it's really a process of learning collaboratives and, really helping disseminate new knowledge out into practice. And that is typically a slow process, but in the case of these categories of medications hasn't been as slow.
I think some of that is direct to consumer.
Some of it is, yep.
And so one of the things, the last question to think about, in case nobody has any from the audience, is should we be doing trials, development, and implementation direct to consumer and just cut out the middleman? Just asking.
Well, I will say this. I think a lot of the adverse effects that we hear reported about GLP-1 receptor agonists are a result of direct to consumer, because I do not see those adverse effects in my hands. But I think when people use the medications improperly, potentially take doses that are too high, without the right sorts of supports nutritionally and other ways, is when you actually see adverse effects. So I'm not convinced that direct to consumer is the solution, although I think having people be informed about what it is that they need is a good thing to do.
Good. Take some questions from the audience if there are any. Raise your hand and somebody will bring a microphone to you. Seeing none, we'll go back to the panel with some pre-prepared questions. It's all solved. So
I think diversity in trials is a really important topic. I actually do a lot of speaking to just audiences of endocrinologists and physicians, and I was presenting the FLOW trial last week, and a black endocrinologist in the audience said to me, "Well, you tell me this works for kidney disease, but how do I know that this is going to work in my black patients?" And so I think having the real partnership, and I think this is where we can also do better on the implementation end, is having the real partnership with our participants to really be in trials. And actually, I think that many companies do a very good job of this. So I'm not trying to, but I think really understanding what it takes for someone to participate in a trial, supporting that effort, and partnering with participants is crucially important.
I think this was one of the reasons that Recursion and Helix actually coupled, is they were going into communities rather than into traditional academic medical centers.
Yeah, I think that's a really important strategy.
I do think that's hugely important, as we said. I think, if you're thinking about omics, you need the extremes. You're not going to get it from looking at the middle of the population.
Yeah.
Lotte.
And how about also, now it's a bunch of cardiologists here, or cardiology knowledgeable people. How about women, right?
Yes,
how about
totally.
the fact that women are not diagnosed properly because the threshold has been set based on studies on men?
I think that's a very good point. And also, people are always, I think, conscious of the risks, the additional risks in treating women. It's one of the biggest challenges, actually, with preeclampsia has been, you're always worried about fetal risk. But again, are those things, I think, that you're seeing as part of early decision-making in drug development, or are they all post hoc 15, 17 years down the line? How are we going to shorten that timeline? Any of you? More money, less money, more drugs, fewer drugs?
One question is, once a drug has been approved, can we accept less stringent outcomes? I mean, maybe this doesn't help you at your early stage, but I think the success of things like, the STEP-HF and STEP-HF diabetes trials, which really looked at an outcome of the 6-minute walk test and the KCCQ, I think for certain things, once you know the safety of a drug, could we look at more intermediate outcomes of efficacy, or does it actually need to be a hard cardiovascular outcome? I don't know.
I suppose one of the things, I think this is what Jason was referring to and Dave mentioned. In oncology, really, you're starting off with efficacy, but with intermediate biomarkers, and then pivoting to therapeutic trials that may be powered adequately. But in cardiology, what we tend to do is really just show early safety, then go to very specific indications, and then only have broader safety trials. So is there a potential to renegotiate the space with the regulators, given what we're seeing? I think a lot of the original hard endpoints in cardiovascular trials were actually driven by adverse effects in one or two areas. And now we're seeing probably disproportionately adverse effects from not getting the right drugs to the right people for a decade after the drug is available. Is there a way to renegotiate that in cardiovascular and metabolic? Dave.
Thanks, Calum. So I guess if we're going back to the oncology analogy, let's not glamorize that too much, right? So we've been giving platinum doublets to non-small cell lung cancer patients for over 50 years and don't know who responds. Right. So I think it's a very big challenge. Plus, most of the tumor shrinkage and PFS data don't correlate with overall survival, nor do we actually run those trials. So I think the benefit in cardiovascular disease is we've got a lot of drugs that work. The problem in oncology is not many drugs that work. Yep. I do think too many drugs get pursued in these indications. I'm not sure how much that contributes to the problem, but the oncology analogy would be, at one point there are 119 CD19 CAR T cell therapy programs being tried. Right. Similarly, how many TTR trials do we need to run? How many novel genetic medicine approaches to TTR do we need to support? And how many, GLP-1, GLP, GIP, et cetera, need to be pursued? I think where you want to see that innovation is, again, matching new technologies to those targets that are very well validated and have proven to do incredible things. What technology platforms and small molecules will unlock novelty here, right? So, Lilly, of course, goes to Chugai for their non-peptidic orals. There's other oral, macrocyclic platforms that are being deployed against these targets now that may provide benefit. And so, where do you want to deploy globally resources? For any individual company, it can make a lot of sense to do that. But as an ecosystem, I'm not sure it gets us where we want to be.
Got it. Well, I think that's a really good point to finish on. I'd like to thank all of the panelists for excellent contributions, and thank the audience for listening.